Okay, let's start it. My talk, first, thanks for the HCA Asian meeting organizer. And it is my great pleasure to share what we did in the cortical development in the human. My title is Spatial and Temporal Regulation for Human Cortex Development, reviewed by single-cell RNA-Seq analyze. My name is Xiaoqun Wang. I from a State K Laboratory of Brain and Cognitive Science, Institute of Biophysics, Chinese Academic Science in Beijing. It's my really pleasure to talk about what we did. Today I'm going to talk about three stories. I will go over very quickly about what we did in the past couple of years. But first, first the story is like what we did on the prefrontal cortex development. As everybody knows, PFC, prefrontal cortex, serves as the center of the higher order cognition functions, such as memory, cognition ability, decision making, and social behavior. The mammalian prefrontal cortex comprises a set of highly specialized brain area containing billions of cells and serves as the center of the highest order cognition function, such as memory, cognition ability, decision making, and so on. Although neural circuits are formed in the later stage of human embryonic developmental and even after birth, a diverse class of functional cells are generated and migrate to the appropriate location earlier in the development. And also this function of the prefrontal cortex contribute to cognition deficit and the majority of the neural developmental disorder. Thus, there's really a necessity need for detailed knowledge of the development of the prefrontal. Thus, this is very important to understand how this part of the brain develop and form. So the first a couple of years ago, with a very fine consent, a letter, and also with a lot of help from the hospital, we would be able to assess the human fetal brain with so what we did is like we try to sampling the cell from three parts of the brain, the prefrontal part of the brain, from three major stages. One is from early gestation from 8 to 13 week stage, and one from a middle early stage. It's like a gestation week 16 to 19. And also we also sample the cell from middle gestation week, about like 23 to 26. So totally, we actually that time we just use like smart-seq technique. We use mouse pipette to pick the cell from unsupervised way to pick up the cell for perform the single-cell RNA-Seq. So totally, we just get like about like 9394 cell. After quality control, we can get about like a 2300 cell. Most of the cell actually we can get the information about the gene average is like it's about like 2654 genes from every each cell. So we also, for sure, we just perform the testing analyze. Actually, we can you can see we can beautifully get the cell from six major class like interneuron, microglia, OPC, neuron protected cell, erythrocyte, and exciting majority is excitatory neuron. And also we can have the time tag with every single cell. You can see from gestation week from 8 all the way to the gestation week like a 26, like this kind of like a dark blue color. So we can just visualize of the major class of the cell use testing, and also which show the 6 main cell types in the prefrontal cortex. So we perform the heat map. Actually, you can show the block of the gene enriched in every each cell type. So we can actually pull out some. We can actually identify some like a new neuronal marker, new molecular marker from a specific like matrix like excitatory neuron, interneuron. We can actually pull out some like a new molecular marker for this kind of like a cell. We also can perform. The hierarchy cluster with random foreign assay and we can further cluster 35 subclass of this cell. Actually, you can see for microglia we can get a 4 subcluster, and for the excitatory neuron we can get a 8 subcluster, and also we can pull out some new marker for like a excitatory neuron, and also some like a SFRP1 for neural progenitor cell for in the human. Further, we do the trajectory analysis by Monocle assay. We can clearly show the developmental stage for the prefrontal. You can see the distance from a cell to the root corresponding to the pseudo-time. The branch trajectory are plotted as a 2-dimension tree layout. You can see from the start point, it's clearly the neural progenitor cell, and they can follow the trajectory to the excitatory neuron. For the OPC and astrocyte actually divert from early beginning, go to another trajectory, right? And you can see OPC and astrocyte share another trajectory. And also from the time tag, you can see clearly from early stage from Jackson 8, majority cell actually from A to, you can see to 10, and from the end point, you can see astrocyte and oligodendrocyte, OPC, majority belong to like a later stage, like Jackson 23 and Jackson week 26. Yeah. We using Monocle analyze, we found that the developmental trajectory of NPC follows 3 major paths. Earlier and later paths from a ventricular zone start point to radio glial cell, like from start point here. You can go to the either intermediate progenitor cell or the later path to the ORD cell path. You can see from here the Hallback scene, which is labeled ORD cell, UNMES, which is labeled IP cell, so they can follow the early path and the later path here. IPC intermediate progenitor cell this transit act as a transit amplified progenitor, which is labeled by EOMES, and enables the rapid generation of neuron during cortical development. To illustrate the intermediate progenitor cell development, we assess the relatively expression of EOMES in the pseudo-time, and the peak during the Jackson week 10 and the Jackson week 16. We found that they have a two peak of the intermediate progenitor cell genesis. To validate this result, we actually performed immunostaining cortical of the prefrontal from Jackson week 8 to Jackson week 19. You can see clearly one wave from early stage is just labeled subventricular zone from here, and the later stage we label all majority in the outer subventricular zone here, which is believed to host a majority of the neural progenitor cell or precursor cell for later for the cortical development in the human. Thereafter, we can find the IPC were not generated actively until from here you can see we observed early increase in the number of IPC at Jackson week 8, just above the sick ventricle zone here. At Jackson week 10, the number of IPC increased rapidly in the expansion of the SVZ. Thereafter, IPC were not generated actively until Jackson week 16, when they are abundant in the ISVZ, inner SVZ, and OSVZ here. So quantification collaborate with the 2 peak of IPC proliferating at Jackson week 10 and Jackson week 16. Primarily, actually, you can find that this is located in SVZ and OSVZ, respectively. These results suggest the accumulated IPC at Jackson week 10 might orientate from the ventricular zone VRG cell, whereas the majority of the intermediate progenitor cell, especially those located in the OSVZ region, might originate from the ORD cell. They are present at Jackson week 16. So by this, we can actually analyze and pull out the origin and the neurogenesis pattern of the intermediate progenitor cell. We then use the iron in situ hybridization to validate these candidates. So for the IPC cell, you can see intermediate protein cell marker. You can see we found like a NHLH1 and NHLH2 and SLC1A3 to PP1R17, which is actually perfectly localized in the S region to reveal the to label the intermediate protein cell. But you also can see some, but their painting and the painting and the localization actually show the diversity which suggests even within the intermediate protein cell, there are also diversity of the cell located there. So last, actually, we use GSE gene-SEA analyze. We can review like exon kinase signaling pathway, which were heavily neurotrophin signaling pathway actually was involved in the excitatory neuron and the interneuron, and the Jetson 16 and the Jetson 20C release their maturation. And we also can review the excitatory neuron mature much earlier than the interneuron. From most of the part, the important thing we pull out is like we found a notch signal pathway, which heavily involve this prefrontal cortex development, especially one of the gene which we notch2, which is actually is a notch2NL gene, which is a human specific gene. We involved mammalian human brain development, which actually when we published this paper in back to 2018, there a couple of months, there are 2 papers come out in Cell support our result. So summary, we analyzed 2,300 single cell and in the development of human prefrontal cortex from gestational week 8 to 26. We identified 35 subtypes of cell in 6 mid clusters and tracing the developmental trajectory of this cell. A detailed analysis of neuron progenitor cell highlights the new marker gene and a unique developmental feature of the intermediate progenitor cell. And we mapped the time. We mapped the timeline of the neurogenesis of excitatory neuron in the prefrontal cortex. And we also reviewed the excitatory neuron became mature earlier than the interneuron. So from this, since we already can perform the single cell analyze to tracing the prefrontal cortex development. So later, next story, we try to understand there are different cell type and molecular feature in different human fetal brain. So we actually try to answer the question of spatial transcriptome survey of human embryonic cerebral cortex by single cell INC. So we actually sampling cell from 22 region. Totally we get 4,213 cell from 22 different brain region, and we actually also can roughly classify this cell into 3 classes: glial cell, neuron, and the non-neuronal cell. And further, we actually can subclass this cell into even more detail in the 29 different subclasses, like excitatory neuron from 1 to 4, interneuron from 1 to 9, and also we also can find like a Malingin cell, B cell, T cell, and also microglia 1 to 3, and also endothelial cell and a blood cell. From all these cell neuron, since we can subclass like excitatory neuron into 8 and interneuron into 8 subclass and the 4th subclass of excitatory neuron. Every each neuron we can see, for example, in the interneuron we can see RHX6 can majority mark interneuron 1 to 4, and CARB2 can identify the interneuron 5 to 8. For the 4th subcluster, you can see there are some share some majority of the gene actually. From this we actually identify from interneuron. You can see they can roughly divide into the CARB2 positive and also RSS6 positive cell. You can see the blue color shows the majority cell interneuron is RSS6 positive cell located in this area, and some like a region actually just focus on there are CARB2 positive interneuron located in this purple region. You can see there are some regional distribution, diversity and heterogenesis. From this different region, especially for the excitatory neuron, we perform time like expression pattern and co-expression network analyze for autism risk gene. From this kind of the high risk autism gene, we actually found excitatory subtype 4 actually expressing all these kind of gene, highly risk autism gene. That might suggest that during the developmental stage, there's some like cell subtype might very valuable to this kind of like autism gene expression. So the last question I will just go over very quickly. And we actually, since we already found the different brain region have different pattern and a molecular feature, so actually then we continue to perform lineage, put in a lineage assay. So we pick the cell from 5 different regions, from frontal lobe, temporal lobe, and parietal lobe, and occipital lobe, and also one from the pons region. We actually follow the whole developmental stage. So we unbiased sampling from just in week 9 to just week 28, and we majority from these 5 regions: FL, PL, OL, TL, and the pons region. And totally we perform SMART-Seq2 and SMART-Seq2 and 6 sequence method, and we pull out like 14,239 individual cell. We actually can get average 4,410 gene, and every gene have every cell have like 120,324 transcripts, and with all these kind of sample were probably considered donated tissue across different time point. So we perform testing, and we can actually also can cell type in the human embryonic and the fetal brain development. We can subclass them perfectly. And actually, we from this part of the research, we found like the earlier development and the maturation of exercise in the pons are much more mature than in the cortex at the same development stage. You can see here a majority this is blue dot is from pons, and also this is from cerebral cortex. Actually, you can see the early developmental in the different other than the cortical region. Which also can be revealed by immunostaining in the cortex and the pons region by this marker like a neuron 2 and GFAP marker. Later, actually, we identify like a 4 subclass of oligoprogenitor and oligodendrocyte in different regions. Actually, we also perform the pseudo monocle analysis. Actually, we found these the oligodendrocyte from different brain region actually developed in quite a similar without some regionalized different pattern. Further, actually, we try to identify the neuron progenitor cell and their developmental in the human cortical region. We found they are quite one of the subclasses of neuron progenitor cell actually shows very unique feature. They partly have a progenitor cell marker, but also partly always show that some like neuronal marker, which suggests it's my transit stage of the neuron progenitor cell in the ventricular lobe. And once we find this specific subclasses marker, we found we specifically analyze this subclass 3 developmental in this human cortical region. Actually, we found like a lot of like specific marker can label and review this cell. Majority cell located in the SVZ region. This is suggests and also highly express Wnt and beta-catenin signal pathway. Further, we found this subtype. We found like link RNA regulated in the ORD cell and the cortical neuron presented cell. We found this one of the link RNA, link 00943, specifically expressed in this ventricular zone and the subventricular zone. And also, once we overexpress this link RNA, actually we can boost the outer radioglial cell genesis and generate more, so we can generate more neuron or with more potential neuron genesis. We further analyze the trajectory of the NPC and pseudo envelope of the NPC development and the maturation. Actually, we found the branch one generate ORD cell, generate ORD cell, and the branch two generate an NPC, which is a majority come from the radioglial cell, which is suggests this new subtype of neuron presented cell generated from the ventricular zone, radioglial cell. Compared to the olfactory lobe, the NPC located in the frontal lobe actually might mature or develop earlier, suggests different brain region might have a different time cost for their development. Yeah. Further, we actually we found excitatory neuron during the developmental stage. We found there are majority of those deep layer neuron and up layer neuron actually we found this kind of neuron also have a very different maturation trajectory in the different brain region. Especially you can see especially you can from here you can find the regional differences in the development of the cortical region, especially for the temporal lobe. They actually have a much more mature maturation of this all kind of excitatory neuron. So summarize, we perform a single cell transcriptome profiling of the full cortical lobe and pons during the human embryonic and fetal development. We identify distinct subtype of NPC and their molecular signature, including a group of previously identified transient NPC. The neuron from clear spatial and temporal distinction, while glial cells of the different origin show developmental patterns similar to these mice. So we captured the development trajectory of oligodendrocyte lineage cell until the human middle fetal state. We also verify regional specified characterization of the neuron in the cortex, including their distinct electrophysiological feature. So different brain region, actually the neuron maturation and the trajectory developmental might have a regional specific feature. So so I would like to stop here to thanks my funding and my very good friend collaborator Fu Tang from Peking University, and also Xiaoqun right now in Guangdong Biland Laboratory, and also Professor Qiao Jie from PKU, and also from Professor Zhang Jun from Capital Medical University. All this work was performed by Qian Wu and Sui Juanzhong and Jing Liu, and also other lab members in my lab. And I would like to stop here to have time to take some questions. Thank you.
Yeah, hey Xiaoqun, I saw audience give a question called
okay, let me pull out where can I find the question? Okay.
Do you think the interneuron are generated in the thalamus? It's a Japanese friend.
No, actually, most I think neuron all generate from the proliferating zone, ventricular zone, and the subventricular zone, and also OSVZ in the human fetal brain. They generate and migrate to the different part of the brain region. Yeah. Let me see. When can I find the question?
Yeah, looking at the question and answers.
Okay. Let me see.
And the second one is Is the NPC3 specifically to particular brain region or age?
APC I think is right now we continue try to analyze that. I think which is the NPC generated from a ventricular zone radial cell. So the majority located in subventricular zone abundantly. So I believe they contribute the majority for the second peak, like that we talk about in the first part. Generate maybe in the first then second peak to help the neurogenesis in the second in the first peak, and also maybe sometime in the second peak. But we can be the most let's see if the NPC specific to particular brain region or age, no. I think they might generate maybe earlier deeply neuron, but not specifically in specific region. Which is we don't know very clear that, but we need maybe perform more immunostimulation for the whole brain part. Let's see. We can be the most potential downstream application of the impressive panel of something. Another question is like, what can be the most potential downstream application of the expression panel of the autism risk gene? That's a very good question. We don't know that yet. I think it's actually a couple of years. Last year, my previous mentor, Arnold Kristen, actually published a Nature paper. They try to they pull out the single cell atlas from the autism patient. I think we should actually definitely compare what we got from the subclass 4 to what we got, the most valuable gene, the cell type specificity. I think we might get some like a similar feature for this kind of specific cell subtype. When is the peak of the neuron genesis in the OSVZ? That's a very good question. Well, OSVZ products actually generate start from the gestation week 16 and might reach the peak at the gestation week 20. And actually, this OSVZ tend to be a shrink and around like a gestation week 22. Yes. When I think I answered this question. So.
Okay.